Okay, so we're gonna pick off where we left off on Tuesday, with the development of animals. Also, today's lab, we will be learning how to uh, transcribe mRNA into protein. Okay, so we're gonna do a few problems, and then I'm gonna assign you a couple of problems um, on Canvas. So you're not gonna use the McGraw-Hill. Your points are gonna be given through Canvas on your answers. Okay, so um, I'll kind of show you that in a little bit. It'll be the exact same way that I gave you the assignment for the studying for the exam. Okay. All right. I think I stopped at echinoderms. Okay, so echinoderms. These are going to be like starfish. Typically, you'll see these organisms found in aquatic environments, so such as starfish, and they do have internal skeletons as well as they can have external skeletons. Okay, and this is why they're more similar to chordates, arthropods, roundworms, annelids, and all those organisms before that have exoskeletons rather than endoskeletons. Okay. So the member of a group of slow-moving stationary marine animals characterized by rough spiny skin and water vascular system, typically an endoskeleton and radial symmetry in adults. Okay. Echinoderms include sea stars, sea urchins, and sand dollars, right? So radial symmetry meaning that you can slice that pie and it's the same slice no matter where you slice it at, right? Um, we have bilateral symmetry we talked about earlier. We are bilaterally symmetric, right? You can divide us in half and then we are even. If you're radial symmetry, think of a cake. If you cut it in half in multiple different locations, then you know that they are the same throughout. They're almost like a circular organism, pie-shaped organism. Okay, so bilaterally symmetrical typically indicates more advanced life form in terms of developmental history, right? If you are cephalized, if you have eyes, a head, a mouth, oriented in a certain directionality, then you're typically more complex than radial organisms, which are sand dollars, starfish, things like that. Okay, radial symmetry indicates primitive. Okay, because again, it's the same throughout. Uh, all right. So the water vascular system. So life originated in water. Okay, and that's very important to understand because if you think about circulatory systems and the transfer of gases in mammals or large organisms, we typically have to think back to our roots and how we originated from the ocean or from liquid water. Okay, water provided us with that early circulatory system, right? Water was able to bathe our tissues and provide gas exchange. Okay, so you'll see this with amphibians. They do have lungs, but they're also able to exchange gases through their tissues. Um, so organisms that are required to be around water, typically you'll see developmentally, they're a little less complex because they're obligated to be in water. And you see echinoderms, this is true as well. The more complex the organism is, um, typically you see the more developed their circulatory system is as well. And you see this in the heart, right? Where you have a four-chambered heart or a three-chambered heart um, in amphibians, okay? And whereas mammals have that four-chamber heart, okay? They just have a more developed um, vascular system, okay? uh, endoskeleton. So a hard interior skeleton located within the soft tissues of an animal, found in all vertebrates and a few invertebrates and echinoderms. Water vascular system. And echinoderms are radially arranged system of water-filled canals that branch into extensions called tube feet. This system provides movement and circulates water, facilitating gas exchange and waste disposal. Right. So this primitive vascular system, this primitive circulatory system, is very important in development. Okay. Um, Being able to have gas exchange in the body system is very important, right? We breathe, we exhale, we bring in oxygen for respiration, and we breathe out the carbon dioxide, which was broken down in that citric acid cycle. Okay, um, Without this circulatory system, we're going to be it's going to be very difficult for organisms to get over a certain size, right? And this is kind of the big thing with insects. Okay, they have a trachea system, right? They do not have a very advanced circulatory system such as we do. Okay, so that therefore they're limited to the size they're able to be. Okay, that's why you don't see spiders the size of a car walking around, right? They can only perform gas exchange on a very small level, so they're limited to about this to the size of their bodies. Okay. Um, Again, they have tracheal systems, which are just little pockets in their body where they can absorb or have gas exchange. Okay. All right. Echinoderms diversity. So we have sea stars. Interesting on how or sea stars actually hunt. They One thing about clams is clams actually don't have, they're very strong and sturdy at closing their shells, and they can hold, the shell closed for a little bit. Sea stars will exploit the dexterity of the clams. So what that means, it'll exploit how long that clam will able to hold its shell shut. Okay, so what a sea star will do is actually climb over the clam, and then it'll start pulling the clam shell open slowly, and it will sit there. Um, and then it'll slowly pull, and then that clam will eventually get tired. It doesn't have good dexterity. It's very strong, but doesn't have good, I guess, stamina is a better word too. So it'll slowly open that clamshell, and then after it will dump its guts and digestive enzymes into that clam, and then it will be able to absorb the clam being digested. So it's quite a nasty predator if you think about it. So imagine being able to dump your guts into a clam and then digesting that um, from the outside in. Okay. So again, mouth will crack in mollusk shell, and it'll dump out its guts kind of into that clam shell. So very interesting. So sea urchins, so spherical, they have no arms. If you look closely, you can see long tube feet projecting among the spines. Typically, we see sea urchin roe or sea urchin eggs being consumed um, by humans. These are also an issue for seaweed, so kelp. Kelp, they can actually consume large amounts of kelp. They'll eat the bottom of the kelp, and then that kelp will be detached from the bottom of the sea floor. And again, they eat a very small portion of the kelp. The kelp is lost from the sea urchin, so they were a problem for a while in California. Oh, excuse me. All right, sea cucumber. On casual inspection, the sea cucumber does not look much like other echinoderms. However, a closer look would reveal many echinoderm traits, including 5 rows of tube feet. So these little projections coming off the body are tube feet. And if you ever felt sea cucumber, you can feel the internal skeleton, the endoskeleton, right? If you push on that sea cucumber, you can feel there's like a bony almost hard interior to that organism. Okay. Okay. Sand dollar. Live sand dollars have a skin of sort, movable spines covering a rigid skeleton. A set of 5 pores arranged in star pattern allows seawater to be drawn into the sand dollar's body. And again, these are more or less filter feeders like porifera. Okay. All right, vertebrate evolution diversity. So all vertebrates have endoskeletons with a skull and backbone. So here you see the skull and you see the spine or backbone with the spinal cord being housed within. Okay. So the snake skeleton, like those of all vertebrates, has a skull and a backbone consisting of vertebrae. Okay. And again, these are interlocking bones, and you have that spinal cord that's protected within uh, um, the spine or within the vertebral column. Okay. Um, so these are all of articulating very closely together. Um, in humans, our spine is a little less movable than snakes, right? We have these different foramen that allow for our nerve roots to be extending out from. Um, yeah. All right. So characteristic of chordates. Oh, thank you, Lucia. chordates. So this is going to be incorporating all of the vertebrates. So that's including birds, reptiles, mammals, anything with a spiny center. So this will include fish as well. So bony fish and even sharks. Even though sharks do not have bone, they have cartilage. Okay. All right. Chordata or chordate. An animal that at some point during its development has a dorsal hollow nerve cord, a notochord, pharyngeal slits, and postanal tail. So remember those pharyngeal slits that we showed with the embryonic development? Okay, that's important. So that's a trait of all chordates. Chordates include lancets, tunicates, and vertebrates. Okay, dorsal hollow nerve cord, one of the four hallmarks of chordates, the chordate brain and spinal cord. So these are going to be housed in the CSN or the central nervous system. Okay, so you often hear that referred to as the CSN. Right, this is going to be the main nervous tissue of the chordates. So notochord is a flexible cartilage-like longitudinal rod located between the digestive tract and nerve cord in chordate animals, present only in embryos in many species. Okay, so again, this is going to be early on in the development of the fetus of many of the organisms in chordate. So it's a notochord. Pharyngeal slit. This is a gill structure in the pharynx found in chordate embryos and some adult chordates. Postanal tail, a tail posterior to the anus found in chordate embryos and most adult chordates. So we do have this postanal tail development. Sometimes you even have individuals that will keep that tail even after being born. So there are some genetic abnormalities that can occur, and people have been born with tail-like protrusions from their spine. Okay, and it can be very painful, right? It's like a bony protrusion with the spinal cord extends into. And again, we've lost the use of the tail, so our coccyx is almost like a vestigial organ, right? So we talked about vestigial organs or vestigial structures. So remember the whale and the bone, the hip bone that they don't need, or the leg bone they don't need anymore, but it's still in their body. Well, our coccyx is a remnants of our tail that we once had. So again, this can be considered a vestigial structure because again, we break our tailbone quite often, right? A lot of humans break their tailbone. It can be more of a problem than a good thing. It also gets in the way of birth. Some people have the protrusion or the tailbone, the coccyx that can get in the way. All right, chordate characteristics. We have that notochord. Okay, remember it's going to be the cartilaginous tube that's going to be housing the central nervous tissue. Pharyngeal slits. Again, these are gill slits. You have that postanal tail, that extension of the spinal cord. Then dorsal hollow nerve root. Again, this is a CSN or central nervous system. Okay. All right. So tunicates, one of a group of stationary invertebrate chordates. Lancet, lancelet, one of a group of blade-like invertebrate chordates, and vertebrate, a chordate animal with a backbone. Vertebrates include lampreys, cartilaginous fishes, bony fishes, amphibians, reptiles, including birds and mammals. So if you didn't know, birds are reptiles. Okay, they are ancient and their deviation, but still reptiles nonetheless. Okay, so invertebrate chordates. So we have lancelet. This is a marine invertebrate. Owes its name to a blade-like shape. Only a few centimeters long, lancelets wiggle backward into the gravel, leaving their mouths exposed and filter tiny food particles from the seawater. Again, so very primitive, very early development, almost looks like a fish, okay? A very primitive fish. So you do have a tail, you do have a mouth. All right, tunicate, a sea squirt, is a stationary animal that filters food from water. These pastel sea squirts get their name from their coloration and the fact that they can quickly expel water to startle intruders. Okay, so I believe the developmental stage or these early chordates are ancestors of some of the less developed chordates. So this looks more like periphery, right? This looks more like our filter feeders periphery. but again, they are single organisms and they're more developed in unison, right? These cells are going to be more closely related together. Whereas, let me see if I can show you what the peripheral is. I know that everyone here in the lecture doesn't know what I'm talking about. So peripheral, right? Oh, here, right? So peripheral is going to be a little less complex than what we're talking about now, okay? So the C squirts are going to be less complex in peripheral, or more complex in peripheral, right? And peripheral, again, are those filter feeders, the sponges. Um, you see there are only a few different cell types here, whereas here, they're going to be a little more developed, okay? All right. So here you have milestones in development of vertebrate. Here we have a vertebral column forming after lampreys and hagfish. These are very interesting organisms, hagfish and lampreys. They do secrete slime. They do provide issues for different types of fish. Hagfish are found in the ocean. And again, these are these organisms that can release large amounts or copious amounts of slime that prevent um, predators from attacking them. So many times they can be attacked. They secrete this gel-like or jelly-like secretion, and then organisms will come to try to attack, and they'll inhale or breathe. I guess they're in water, so the gel-like substance will get stuck in their gill slits, and sometimes they can actually die and prevent them from being consumed. So very interesting mechanism for protection. And again, this is a very ancient lineage of fish. Lampreys, you see these, there's a problem in the Great Lakes. Lampreys have been attaching or attacking fish in the lake. Again, lampreys are kind of interesting in the fact that they're parasitic. So they latch on the fish and let me see, they latch on the fish and they can cause some issues for fish. Let me see, I want to show you. I feel like I'm not doing it justice. So hagfish are kind of gross. I guess that's a positive thing about having. So again, these are going to be found in the ocean, and they have these jaws. So it's not true jaws, but they actually can attach. They typically eat dying and decaying uh, sea or organisms that have fallen to the bottom of the ocean, and they'll come and they'll attach, and they'll wrap themselves in a knot and rip chunks of tissue out. So they don't really have jaws to latch and bite and remove the tissue. They need to perform this awkward kind of knot tying and ripping of the dying, decaying plant matter. So here's an example of the slime that they can produce when stressed. Okay, and again, it's very thick, and organisms can get that caught in the lungs, and then it can be detrimental to that predator. Okay, it can prevent them from being attacked. Okay, so these are very interesting organisms. Okay, so these are hagfish. Okay, and then you do have the lampreys. So again, another jawless fish. These are parasitic though, so they can latch onto fish like you see here. Um, Actually, I don't think those are hagfish. I think those might be. Oh no, I think that is a hagfish. There's a fin right here. I thought they were eels or leeches. But again, you do see that they have eyes. They have this raspy uh, protrusion at the front, so it almost looks like a mouth, but it's not. And again, they can attach to the fish, and they have this scraping jaw component, which actually rubs into the fish and cuts a hole into the fish, and then they can essentially suck out the blood. Okay, so this has been a problem in the Great Lakes region where these fish have been really draining the local fish population. So you see here, they can cut holes into the fish that are found there, right? Into the trout or the salmon or whatever organisms that they're going to be attaching to in the fresh water. Okay, so they are providing a problem for those lakes because they're cutting into the commercial fishing market or the fishing market in general, where individuals would like to be fishing, but they're getting a lot of these fish with holes cut in them, and it's not desirable. Okay. So yeah, I thought I'd show you some gross pictures of interesting fish. Oops, wrong one. All right. Just wanted to make sure I did that justice because I know those are interesting looking organisms. Okay, so then you have the jaws. So after these jawless fish, again, would be considered an outgroup to these organisms here in this phylogenetic tree. We have jaws. So cartilaginous fish. So this is sharks. Sharks are more primitive when in comparison to a bony fish in terms of development and evolution. We have legs in amphibians, and then amniotic egg in reptiles and mammals. Okay, whereas amphibians lack those amniotic eggs. Okay, so they lack that soft shell and um, the yolk and all that that reptiles and mammals have. Okay, so fishes, we have hagfish, lampreys, cartilaginous fishes, and bony fishes. Okay, cartilaginous fish, a fish has flexible skeleton made of cartilage, lateral line system. This is a row of sensory organs along each side of a fish's body. Sensitive to changes in water pressure, it enables a fish to detect minor vibrations in the water. Okay, so you've seen this lateral line system in fish before, where they have that almost like that sixth sense in water, where they can detect movement or electrical signals. I'm sure everyone's seen the sharks or the documentary on sharks. They do have these electrical sensors in the front of their faces or in front of their jaws, where they can actually detect electrical impulses of fish, right? So again, this is going to be similar to the lateral line system, where it's a sensing organ that can detect minute changes and pressure changes in the water. Okay, so bony fish, a fish that has stiff skeleton reinforced by calcium salts. These calcium salts are just referring to the bone. Uh, Operculum, this is a protective flap on the side of a bony fish's head that covers a chamber housing the gills. This is very important for differentiating cartilaginous fish, so rays, sharks, in comparison to bony fish. All right, this operculum is going to be especially important because one of the main things about sharks that we see that fish don't have to do is continuously swim. Right, fish can float and sit and actually use this operculum to allow for water to be passed and moved over their gill slits. Okay, whereas typically sharks or cartilaginous fish require movement and require a constant states of movement. So you'll see that, right? They'll say, "Oh, don't stop that shark from swimming. You need to continuously put water over their gills because they do not have the ability to pass water over their gills unless they're swimming." Okay. There are exceptions to this rule, right? There's, I think it's nerf sharks, and certain rays can suck in water with their jaws, but again, they do not. They lack the operculum. They do not have any type of bone or cartilage moving water physically over their gills. They need to be actively doing something with their jaws in order to get fresh salt water over their lungs to perform gas exchange. Okay, so that's the difference between the bony fish and the cartilaginous fish. One of the differences, right? One of the major differences is that operculum. Okay, fishes do have swim bladders, a gas-filled internal sac that helps bony fish maintain buoyancy. Sharks have a fatty liver, okay, and they constantly swim. If they don't swim, they sink typically. Okay, so ray fin fish, a bony fish in which fins are web of skin supported by thin, flexible skeletal rays. All but one living species of bony fish are ray fins. Okay, so lobe fin fish, a bony fish with strong muscular fins supported by bones. Okay, so here we have amphibians. Okay, so this is going to be that bridge between life on land and life on the ocean. Okay, so we do have frogs and salamanders, the two major groups. Again, one of the major things with amphibians and frogs, they do have lungs, right? They can perform gas exchange with the air, but they can also perform gas exchange with their skin. Okay, okay, so they can have oxygen or gas exchange with water through their skin. So they need to be moist or wet constantly. Okay. There are frogs that can live in desert like environments, but again, they do have to be closely associated with water still. Right. So here's frog eggs. And these are obligate, these frog eggs need to be in water. Okay. So it's one of the major key things with frogs. Okay. They need to have some sort of water system even to lay their eggs, right? Because of tadpoles. One of their life stages is going to require water. Okay, so you have a tadpole again, then you have that changing or metamorphosis into a frog eventually. But tadpoles do require water. Okay. All right. So lobe-finned fish, fossils of some lobe-finned fishes have skeletal supports extending into their fins. Early amphibians, fossils of early amphibians have limb skeletons that probably function in helping them move to land, move on to land. Excuse me. Okay. So again, this is going to be the growing or the extension of their fins into actual feet. Excuse me. All right, reptiles. So reptiles again are going to be descendants of amphibians. You're going to have a couple types of reptiles. You're going to have uh, endoderms and exoderms in terms of their body regulation, and then you'll have birds with feathers, and then you'll have reptiles with scales instead of feathers. Okay. So a member of the clade of amniotes that includes snakes, lizards, turtles, crocodiles, alligators, birds, and a number of extinct groups, mostly including most of the dinosaurs. Amniote, member of a clade of tetrapods that has an amniotic egg containing specialized membranes that protect the embryo. Amniotes include mammals and reptiles. Amniotic egg, a shelled egg in which an embryo develops within a fluid-filled amniotic sac and is nourished by yolk. Produced by reptiles and egg-laying mammals, it enables them to complete their life cycles on dry land, right? So this is very important, this amniotic egg. It prevents that life stage that requires a water-dwelling young, okay, that frogs have, right? This amniotic fluid, this amniotic egg, allows for the full development of uh, the uh, the young into functioning land-dwelling organisms. Okay, reptile diversity. So here you have that amniotic egg. Again, snakes have eggs, crocodiles, birds, and then of course dinosaurs. All right. So you see the different variations among um, the reptiles. So you have a Gila monster, a desert dweller of the Southwest, is the only venomous lizard native to the United States. The other one is the Komodo dragon. Okay, crocodile found throughout Central and South Africa. The Nile crocodile can grow up to 20 feet long and weigh nearly 2,000 pounds. So huge, very ancient reptile as well. Um, so again, birds are considered reptiles as well. Their feathers are going to be analogous to the scales that these organisms have, these reptiles have. Okay. Ectotherm. So an animal that warms itself mainly by absorbing heat from its surroundings. So these are the majority of reptiles. So think of your lizards in your backyard. Think of crocodiles, snakes, right? They require warm weather in order to heat their bodies up and run or perform quick tasks, right? So one of the things you can see in the morning is sometimes you'll catch a lizard out trying to sunbathe just before the sun comes up. And what you'll notice is that lizard lacks the ability to get away. Okay. And this is kind of an important trait that deviates or separates birds from non-bird reptiles. Okay. They're ectotherms. They require a heating process in order to get their body up to speed, right? But this also can be a beneficial thing, right? This can provide a means of slowing their metabolism down, right? Crocodiles can go for long periods of time without eating, whereas us and birds cannot, because again, we have to heat our bodies. But again, it can also provide it can also provide a problem for the organisms that are not warmed up, right? So if you ever walked out, you caught a lizard before the sun is actually out, you can see how slow they move. Many times you can walk up to a lizard if it's really cold in the morning and they're out trying to sunbathe, and you can actually pick that lizard up. It will not be able to do anything. If you hold that lizard for a couple minutes in your hand, that lizard would take off in 2 seconds, right? Your own body heat will heat that lizard up at adequate levels to where it can take off at a quick rate, right? But it needs to be, and that organism needs to be heated up in order for this to occur. That heat is required for the muscle tissue and the enzymes to be functioning properly. But this is the same with large reptiles as well, including alligators or crocodiles. If it's cold, they don't move. Okay, it's hard for them. Birds, some members of a group of reptiles with feathers and adaptations for flight. So endotherm, an animal that derives most of its body heat from its own metabolism. So again, endotherms can heat themselves. Birds can heat themselves. One of the downsides to this is that they do have to constantly consume food. Okay, they have to utilize more energy. Whereas a snake can eat once in 3 or 4 months. Uh, we have to consume constantly, right? Birds consume constantly. Think of a hummingbird. A hummingbird eats its own weight in nectar. It has to, because it's constantly moving. It has to keep up its temperature as well. Okay. All right. So the aerodynamics of a bald eagle in flight, both birds and airplanes owe their lift to changes in air pressure caused by the shape of their wings. Okay, so again, these feathers of these reptiles provide, excuse me, a high and low pressure dynamic that's going to allow them to have lift. Okay, allow them to fly. All right, last little topic: mammals. So a mammal, member of a class of endothermic amniotes that possesses mammary glands and hair. Okay, so mammary glands means that we feed our young milk. An egg-laying mammal, such as a duck-billed platypus, is a monotreme. A placental mammal is most mammals the organ that provides nutrients and oxygen to the embryo that helps dispose of its metabolic waste. So the placenta, also called, well, it can be referred to as a chorion as well. So the attachment and then eventual transfer of nutrients to that fetus. The placenta is formed from embryonic tissue and the mother's endometrial blood vessels, right? So think of the egg fertilizing in the fallopian tube in humans and then being transferred down to to the uterus, and then the implantation of the chorion into the uterus, allowing for that transfer of nutrient and gas exchange. Okay, so we have marsupial. This is a pouch mammal such as a kangaroo, opossum, or koala. Marsupials give birth to embryonic offspring that complete development within housed while housed in a pouch and attached to nipples in the mother's abdomen. Right. So you do see marsupials, and you see they're young, and they almost look like little beans. Right. They're very, very small, and the marsupials can house these organisms in their own pouches for a little while until they develop. Okay, and in those pouches, they do have mammary glands. They do have nipples or breast-like tissue that's going to provide them milk. Eutherian, some mammal whose young complete their embryonic development in the uterus, nourished via mother's blood vessels in the placenta, also called a placental mammal. Okay. Placental mammals, mammals whose young complete their embryonic development in the uterus, nourished via the mother's blood vessels in the placenta, also called Eutherian, right? Okay, so here are some mammal diversity. So we have monotremes, duck-billed platypus with the young. These actually look kind of cute. We look closely, there's like a little bill, and then you see little tails. Again, so only mammals that lay eggs are the duck-billed platypus. Okay, so monotremes. There are mammals that lay eggs. So they do have fur, they do have milk, but they lay eggs. So they're still obligated to that egg laying that we see with reptiles and birds. Okay, marsupials. So they're embryonic at birth. So think of these organisms as almost premature when they're born, right? Young marsupials need to have that extra care. They need to be housed in a pouch. They also need to be nourished by milk as well in the pouch. Okay, so they do have nipples or mammary tissue inside of these pouches. So you see this joey or this small kangaroo here. Probably should be out of the pouch by now. It's getting a little old. So in eutherians, so placental mammals, young develop within the uterus of the mother. Uh, they're nurtured by the flow of blood through the dense network of vessels in the placenta. This network full is coated by remnants of the placenta. Okay. And again, one of the things that placental mammals have in comparison to marsupials is their young are younger, more developed. Okay, they're more able to complete different tasks. Oops, in comparison to marsupials or monotremes. But there's a downside to this as well. There's much more energy involved with developing the young here. Okay, whereas marsupials and monotremes, you lay an egg, you literally dump out whatever you need to dump out with that organism, and if it survives, it survives. Marsupials, not a lot of genetic or not a lot of energy is going to be used in that initial birthing of the organism. Much of the energy is going to be required after the developmental stage. Whereas here, the mother is more at risk because of how developed this organism is. But the more developed the young is, the more likely that they will survive. Okay, so you have a higher chance, higher energy investment for single organisms. Okay, whereas here, less energy is invested. Therefore, um, you know, potentially the loss of the young is less detrimental to the organisms or to that species, right? Whereas here, if you lose the child or you lose the young early on, you lose a lot of energy. You lose a lot of stored or invested energy in motherhood. Okay, all right. So that's it for the lecture. So just a quick lab. I'm going to be uploading a problems for this. I know we really didn't go over translation um, of mRNA to proteins. So think of this as a tRNA. Okay. So again, this is essential dogma. We have DNA that's converted into so transcription, so DNA into RNA, then translation. We have the RNA converted into polypeptides. Okay, so again, this is just displaying the central dogma, the initial start of from DNA to RNA to protein. Okay, so if you remember, we have this chart here, um, and this is going to be the anticodons or the codons that are going to be used by tRNA to recognize these different amino acids, right? So these 21 different amino acids. Remember, methionine is always going to be that first amino acid, that AUG, that start codon. Okay, so you'll see this at the very front of a protein, okay, typically. All right. So for today's lab or today's kind of task that I want you to do over the weekend, is I'm going to be giving you some problems and uploading assignment online. And what you're going to do is actually perform or map out some proteins from an mRNA. Okay. And I want you to I'm going to ask you some questions based on that. Okay. So a lot of times it'll be tricky. I'll ask you the AUG or the start versus the stop codon. So I want you to be able to identify that. We didn't really talk about directionality with the nucleotides or the mRNA, but it's typically it's a 5' end that's going to be plugging into the ribosome. Okay, so the 5' end is the side with the phosphate. Okay, so I guess you don't really need to know the directionality. You just need to know. Well, you do need to know the directionality. No, it's okay. We won't do that right now. We'll talk about that next time. But you should be able to identify that there are redundancies within the codon, right? There are multiple codons that can give you one amino acid. This is kind of a protection for conservation of that amino acid, right? If there's a mutation here, you'll still get proline, right? Because there's all four of these different amino acids or different bases here will give you proline, right? Cytosine, cytosine, uracil, cytosine, 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 adenine, cytosine, cytosine, guanine. Remember, in RNA, uracil or thymine is replaced by uracil, so you do not see any T's in the RNA. Okay. All right, genetic code. So the set of rules giving the correspondence between nucleotide triplets, codons, and mRNA, the amino acids and proteins. Again, you have those tRNA molecules that are going to be adding those different amino acids to the elongation during the processing of the ribosome. Okay, so the mRNA comes in, binds to that ribosome, and then you have that tRNA that's going to recognize those different triplets or those different codons. Okay, and that's important, right? That's going to allow for the elongation of that polypeptide strand. Okay, RNA polymerase, this enzyme that links together the growing chain of RNA nucleotides during transcription, using DNA strand as the template, right? That was shown earlier. Okay, so here we have transcription, and then eventually we'll have translation, right? So transcription, you have the RNA polymerase coming, creating the mRNA. So here we have the mRNA. Then the mRNA is going to be transferred to a ribosome, and that ribosome again will then make that protein. Okay. All right. So here's a couple of little problems here that I want you to use or perform. Right. So I want you to write down this. Well, we'll do it together. So here, I'll move this down here. It's a little bit easier. And this is essentially what you're going to be doing for homework. Okay. So it'll be a little assignment with some questions at the end of it. Okay. And again, this is going to be on Canvas. So this is going to be like the last assignment, which I'll show you in a minute. I don't think a lot of people were doing the assignment. So UUU will give you phenylalanine. So you can write PHE, right? Phenylalanine. What else? UCG. So we can look here. U, C, and then G. UCG. Serine. Okay. GGU. GGU is glycine. All right. CCU. Proline. All right, and then we have UCG. UCG. It's going to be serine again. Oops. UCG. UGU, wait. UU, 1, 2, 3, 4, 5. All right, UGU. So we have UGU, cysteine. Okay. And then we have GGA. It's glycine again. All right, and what's the problem here? What do you notice that is different with this polypeptide? So this is going to be the polypeptide. It does not have AUG start. So that's a problem. Okay, it also doesn't have a stop codon. All right, so genes also require the stop codons. But again, I'll ask you those questions later on the assignment. So this is all you're going to be doing. You're going to be using this. You're going to be using the this chart in order to make a polypeptide. And I'm going to ask you a couple of things about the polypeptide. I may ask you about the tertiary, primary, secondary structure. And again, this is just going to be a protein lab, right? This is going to be the early, it's like pre-enzyme understanding, right? Because enzymes are proteins. You should be able to understand or identify certain issues that you find here. So AUG, it's always going to be methionine. Remember, methionine is always going to be the first amino acid in a protein sequence, right? Because this AUG is a start codon, right? So we have UAG. So UAG is stop, okay? So you should be able to identify that, right? Nothing will happen after this. So methionine, and then you have UAG as a stop codon. So here we have a mutation. Something occurred where it produced a mutation, because clearly this is the start of a gene, but you don't transcribe the gene. Something happened there. But you should be able to identify this. So you can say issues here are possibly or a possible mutation. Okay. Right, because you just have a start and then a stop. Okay. Again, methionine, GGG. Here we have GGG, we have glycine, AU, I think it's aspartic acid, AGG, am I missing something? Oh, arginine, and we have CCG, proline, proline. And then we have CUU, CUU, leucine, UAG. Oh, then we have stop. Okay, and CCG is not. So here we have, it looks good to me. So we have a start and a stop codon, okay? So I'm gonna have you do a couple of problems. You're just gonna write down that, that homework on Canvas, okay? So let me just go to Canvas real quick. Hang on, chat. Oh, did I mess up? I might have messed up. What is it? UAG. Oh, it's UGA. Oh yeah, there's redundancies in the stop codons too, Lucia. But good looking out. So there are multiple stop codons. So you have UAG, UAA, UGA. Those three are gonna be stop codons. So, but good looking. So way to understand that. That's important too, because there are multiple stop codons. Yeah. And then we'll talk about frame shift or different mutations as well. I know we haven't really talked about mutations too much, even in genetics, but we'll talk a little bit about mutations and issues with this. Okay. So I know it probably would have been better to explain this during genetics, but I feel like everyone was getting a lot of information thrown at them at that one time. There's a lot of confusion. So I just wanted to take the time to really explain this process and how it works, right? So DNA to RNA, and then we can get our protein. Now that you understand kind of how protein works and how DNA works, how genetic information is stored, I think it's a good time now to kind of show you how to do this and for you to kind of understand how proteins are going to be made, right? And if you remember, this is going to be the transcription or that addition of that transfer RNA or tRNA, making those proteins. Okay. Let me see. So this assignment will be uploaded in, I guess the assignments. I'll probably have it uploaded here. So I know the paper topics and then the essay questions. Let's see. So yeah, it's due the 23rd. So hopefully everyone got this done before the exam, or will be get this done before the exam. Yeah. But again, you only have a few submissions. So just remember to access this on Canvas. So this is why I'm putting this in here, right? It will be in the assignments, but it's much easier to find it on the module since you have so many other assignments. So just go to the modules and go to this last week, right? And then you have exam essay questions, right? So everyone that's watching this, please just go to Canvas. I'm gonna upload this assignment in week 11. Okay, so it'll be translation assignment. Okay, and then it'll be very similar to this. So I'll have that chart posted here, that image of I'll have this chart posted. Okay, and then I can post this PowerPoint too, even though this is just a quick PowerPoint. You have all that information still, and I'm just gonna have you perform those different questions. Okay, and write them in. You can just write them in and do them really quickly, and I'll ask you a couple questions at the end. And I think I'm gonna make it 10 points. I think it'll make it 10 points. It'll be just an little extra points for the assignments. Okay, so just get this done. It's not gonna be too difficult, right? Just follow what I did on the PowerPoint and what I showed you right now, and you should be fine. Okay. All right. Well, that's it for today. I think I went a little over. Oh no, I'm at about an hour, so I'm good. Okay. So next week we'll go. I think it's chapter. I think it's chapter 21. We're jumping forward to animal structure and function. So we're getting into enzymes, digestion, more of the interesting stuff. So larger, big picture things of the body rather than specific DNA stuff, which I know everyone thinks is boring. So we're going to talk about human body, maybe some disease, things like that. So and functions of the body. So it should be a little more interesting for everyone. Um, so yeah. All right. I will. I have office hours right now, so I will see everyone later.